The joy of learning biology, right here online for you and me. Explore the wonder with BioClass.
Excitation of chlorophyll by light. When a chlorophyll molecule absorbs light, it becomes excited, meaning its electrons gain energy and move to a higher energy level. However, this excited state is unstable, and the molecule eventually returns to its lower energy state, releasing the absorbed energy. This energy can be transferred in two ways. Energy transfer. The excited electron passes its energy to another chlorophyll molecule. Electron transfer. The excited electron itself moves to a different molecule, known as an electron acceptor, leaving behind a positively charged hole in the chlorophyll molecule. Photosystems are groups of pigments and proteins found in the thylakoid membranes of chloroplasts that play a central role in photosynthesis, specifically in the light-dependent reactions. There are two main photosystems, Photosystem I and Photosystem II. As we start with Photosystem I, leave a comment in the comment section, suggest what we should cover next, and what we can do to fulfill your needs. Photosystem I. The reaction center of Photosystem I is called P700, which is a special type of chlorophyll molecule. It is named P700 because it absorbs light best at a wavelength of 700 nanometers, made up of the antenna complex and the reaction center. Photosystem I is mainly found in the stroma lamellae of the thylakoid membranes. When light is absorbed by pigments in Photosystem I, the energy excites electrons in P700 to a higher energy level. These excited electrons are captured by a primary electron acceptor, then transferred to ferredoxin. From there, the electrons are transferred to oxidist NADP, along with hydrogen ions from the photolysis of water to form NADPH. This reaction is catalyzed by NADP plus reductase. The electrons lost from Photosystem I reaction center creates an electron hole, which is filled by electrons from Photosystem II. This is located in the thylakoid membranes of the chloroplast, made up of light-harvesting complexes, pigment molecules, chlorophyll pairs, reaction centers, and the primary electron acceptor. When light strikes Photosystem II, electrons in the chlorophylls get excited and move, along the reaction centers, to a higher energy level. They are then taken up by the primary electron acceptor. It's the movement of electrons in the electron transport system that generates the energy. Loss of electrons creates an electron hole, which has to be replaced. This causes the splitting of water in the reaction known as photolysis. This releases electrons to stabilize Photosystem II. The oxygen is given out as a byproduct. The hydrogen ions given out combine with electrons from Photosystems I and NADP+ to form NADPH, a key reducing agent used in the Calvin cycle to make glucose. Photophosphorylation. This is the process by which ATP, adenosine triphosphate, is formed using energy from light during photosynthesis. It takes place in the chloroplasts, specifically in the thylakoid membranes, and involves the use of light energy to drive the addition of a phosphate group to ADP, adenosine diphosphate. Types of photophosphorylation, cyclic photophosphorylation, involves only Photosystem I. No NADPH is produced. ATP is produced. Water is not split, so no oxygen is released, occurs when oxidized NADP is not available. Non-cyclic photophosphorylation involves both Photosystem I and Photosystem II. The electron from Photosystem II travels through an electron transport chain to Photosystem I and then to oxidized NADP to form NADPH. Water is split to replace lost electrons in Photosystem II, produces ATP, NADPH, and oxygen. This is the main pathway during photosynthesis, non-cyclic photophosphorylation. Light simultaneously strikes both photosystems to cause excitation and loss of electrons. Light absorption in photosystem. 2. Light energy is absorbed by chlorophyll molecules in Photosystem II. This energy excites electrons in the reaction center P680, raising them to a higher energy level. The excited electrons are captured by a primary electron acceptor, phospholysis, and then passed to plastoquinone, PQ, leaving P680 positively charged photolysis of water. To replace the electrons lost by P680, water molecules are split in the thylakoid lumen into hydrogen ions, electrons, and oxygen. The electrons replace those lost by P680. The hydrogen ions help in forming NADPH and ATP. The oxygen is released as a byproduct. Electron transport chain. The excited electrons from Photosystem II move through a chain of electron carriers, including plastoquinone, PQ, the cytochrome complex, and plastocyanin, PC. As the electrons flow through the chain, their energy is used to pump hydrogen ions across the thylakoid membrane into the lumen, creating a proton gradient, as the protons flow back into the stroma through the ATP synthase enzyme. Energy is provided which combines ADP to inorganic phosphate to form ATP in what is known as chemiosmosis, light absorption in Photosystem I. Light energy is absorbed by pigments in Photosystem I, exciting electrons in P700 to a higher energy level. The excited electrons are accepted by ferredoxin, FD, leaving P700 positively charged. Replacement of electrons in Photosystem I. The electrons lost by P700 are replaced by electrons arriving from Photosystem II through the electron transport chain. Thus, the flow of electrons continues from water to Photosystem II, then to I. The electrons from ferredoxin are transferred to oxidized NADP, along with hydrogen ions from photolysis by the enzyme NADP reductase, forming NADPH. These hydrogen atoms used here are the ones obtained from the splitting of water. This NADPH provides the reducing power needed for carbon fixation in the Calvin cycle. Watch my next lesson on dark reactions of C3 and C4 plants, end products. At the end of non-cyclic photophosphorylation, ATP is produced for energy, NADPH is produced for reduction reactions, and oxygen is released as a waste product. Non-cyclic photophosphorylation yields equal amounts of ATP and NADPH. However, the dark reactions use more ATP than NADPH, which rapidly depletes faster than NADPH. The excess NADPH short-circuits the non-cyclic photophosphorylation to activate cyclic photophosphorylation, light absorption, in Photosystem I. Light energy is absorbed by pigments in Photosystem I. This energy excites electrons in the reaction center chlorophyll, P700, to a higher energy level. The excited electrons are captured by a primary electron acceptor, then pass through a series of electron carriers, including ferredoxin, FD, and the cytochrome complex, ATP formation. As the electrons pass through the cytochrome complex, some of their energy is used to pump hydrogen ions across the thylakoid membrane into the thylakoid lumen. When hydrogen ions move back into the stroma through ATP synthase, the energy released is used to convert ADP and inorganic phosphate into ATP. After losing energy, the electrons return to the P700 reaction center of Photosystem I. Do not forget to leave a comment. Thank you for watching.